Welcome back, ladies and gentlemen. So, just sort of take a step back and review the big picture of photosynthesis. This diagram actually does a really nice job of showing where the major reactants go in, where the major products get produced, and where those two major components or two major processes of photosynthesis take place, right? So we use that photosystem 1, photosystem 2, those electron transport chains, right? These are those light reactions. They take in the sunlight energy from the sun, water, and ultimately produce us oxygen that comes off as a byproduct, as well as producing ATP and NADPH that then are shuttled over to the Calvin cycle, right? So these guys take place in those thylakoid membranes. The Calvin cycle takes place out in the stroma of our chloroplast, uses that ATP and NADPH energy, right? So we've harnessed the energy from the light, converted into chemical energy that the cells can then use, and move that over to the Calvin cycle, where we take in that carbon dioxide, ultimately fix it into those G3P molecules, regenerate that RUBP, as well as producing recycling ADP and phosphate and NADP plus back over to the light reaction so that they can keep working. And then that extra G3P that gets produced through that carbon fixation in the Calvin cycle can then get used to build things like glucose, which can then be used for cellular respiration in plants, so they can get shuttled over to their glycolysis, Krebs cycle, their oxidative phosphorylation. They can also use it to build cellulose for their cell wall components. They can store it in starch. They can also use it to build any other organic compounds that they need. So in addition to being able to memorize the chemical reaction for cellular respiration and where those products get used and produced and where those things take place, we're going to do the same thing here for photosynthesis. So you should be able to recognize what the chemical reaction is, where those reactants are either created or produced, and then where those things take place in the cell itself. So let's kind of work through this together, right? So if I take my overall photosynthetic reaction, I've got 6 molecules of carbon dioxide that go in as a reactant, as well as 6 molecules of H2O, so 6 molecules of water, plus that sunlight. All right, and that's ultimately going to produce, once we go through those light reactions and the Calvin cycle, I'm going to produce a single molecule of glucose, so C6H12O6, plus 6 molecules of oxygen. Okay, so let's take those reactants and products and let's list them down here in the table. So here's my carbon dioxide, my water, my sunlight, and then the glucose and the oxygen. Okay, so those first 3 there, the carbon dioxide, the water, and the sunlight, are all reactants. So where do they get used? Where do they get put into photosynthesis? Carbon dioxide, right, gets used in the Calvin cycle. H2O is going to be used in the light reactions. Sunlight is also used in those light reactions. And then we have those products, the glucose, the C6H12O6, as well as the oxygen. So where do those get produced? Well, the glucose comes out of the Calvin cycle, really in the form of that G3P, but can then get put together to make that glucose. The oxygen comes out in your light reactions. Okay, so where do these then take place? Well, they all take place in your chloroplast, but let's be a little more specific than that. So when I look at my Calvin cycle, that carbon dioxide, that's going to be taking place in my stroma. All right. The light reactions, take place across the thylakoids. Reactions then take place across the thylakoids, the thylakoid membrane. So that's where I'm going to use both the water as well as the sunlight. And then I produce the glucose in the Calvin cycle. That also takes place again in the stroma. And that oxygen then gets produced in those light reactions again in the thylakoid.
Photosynthesis. It is not some kind of abstract scientific thing. You would be dead without plants and their magical, nay, scientific ability to convert sunlight, carbon dioxide, and water into glucose and pure, delicious oxygen. This happens exclusively through photosynthesis, a process that was developed 450 million years ago and actually rather sucks. It's complicated, inefficient, and confusing, but you are committed to having a better, deeper understanding of our world, or more probably you'd like to do well on your test, so let's delve. There are two sorts of reactions in photosynthesis, the light-dependent reactions and the light-independent reactions, and you've probably already figured out the difference between those two, so that's nice. The light-independent reactions are called the Calvin cycle. Yes, that Calvin cycle. Photosynthesis is basically respiration in reverse, and we've already covered respiration, so maybe you should just go watch that video backwards. Or you could keep watching this one. Either way, I've already talked about what photosynthesis needs in order to work: water, carbon dioxide, and sunlight. So how do they get those things? First, water. Let's assume that we're talking about a vascular plant here. That's the kind of plant that has pipe-like tissues that conduct water, minerals, and other materials to different parts of the plant. These are like trees and grasses and flowering plants. In this case, the roots of the plants absorb the water and bring it to the leaves through tissues called xylem. Carbon dioxide gets in and oxygen gets out through tiny pores in the leaves called stomata. It's actually surprisingly important that plants keep oxygen levels low inside of their leaves for reasons that we will get into later. And finally, individual photons from the sun are absorbed in the plant by a pigment called chlorophyll. Alright, you remember plant cells? If not, you could go watch the video where we spend the whole time talking about plant cells. One thing that plant cells have that animal cells don't: plastids. And what is the most important plastid? The chloroplast, which is not, as it is sometimes portrayed, just a big fat sack of chlorophyll. It's got complicated internal structure. Now, the chlorophyll is stashed in membranous sacs called thylakoids, and the thylakoids are stacked into grana. Inside the thylakoid is the lumen, and outside of the thylakoid, but still inside of the chloroplast, is the stroma. The thylakoid membranes are phospholipid bilayers, which, if you remember, means that they are really good at maintaining concentration gradients of ions and proteins and other things. This means keeping a concentration higher on one side than the other of the membrane. You're going to need to know all of these things, I'm sorry. Now that we've taken our little tour of the chloroplast, it's time to get down to the actual chemistry. First thing that happens, a photon created by the fusion reactions of our sun is about to end its 93 million mile journey by slapping into a molecule of chlorophyll. This kicks off stage one, the light-dependent reactions, proving that yes, nearly all life on our planet is fusion powered. When chlorophyll gets hit by that photon, an electron absorbs that energy and gets excited. This is the technical term for electrons gaining energy and not having anywhere to put it, and when it's done by a photon, it's called photoexcitation. But let's just imagine, for the moment anyway, that every photon is whatever dreamy young man 12-year-old girls are currently obsessed with, and electrons are 12-year-old girls. The trick now, and the entire trick of photosynthesis, is to convert the energy of those electrons into something that the plant can use. We are literally going to be spending the entire rest of the video talking about that. I hope that that's okay with you. That first chlorophyll is not on its own here. It's part of an insanely complicated complex of proteins and lipids and other molecules called photosystem 2 that contains at least 99 different chemicals, including over 30 individual chlorophyll molecules. This is the first of 4 protein complexes that plants need for the light-dependent reactions. And if you think it's complicated that we call the first complex photosystem 2 instead of photosystem 1, then you're welcome to call it by its full name, which is plastoquinone oxoreductase. Oh, no, you don't want to call it that? Right then, photosystem 2. Or, if you want to be brief, PS2. PS2 and indeed all of the protein complexes in the light-dependent reactions straddle the membrane of the thylakoids in the chloroplasts. Now that excited electron is going to go on a journey designed to extract all of its new energy and convert that energy into useful stuff. This is called the electron transport chain, in which energized electrons lose their energy in a series of reactions that capture the energy necessary to keep life living. So PS2's chlorophyll now has this electron that is so excited that when a special protein designed specifically for stealing electrons shows up, the electron actually leaps off of the chlorophyll molecule onto the protein, which we call a mobile electron carrier because it's a mobile electron carrier. The chlorophyll then freaks out like a mother who has just had her 12-year-old daughter abducted by a teen idol and is like, what do I do to fix this problem? And then it, in cooperation with the rest of Photosystem 2, does something so amazing and important that I can barely believe that it keeps happening every day. It splits that ultra-stable molecule, H2O, stealing one of its electrons to replenish the one it lost. The byproducts of this water splitting, hydrogen ions, which are just single protons, and oxygen. Sweet, sweet oxygen. This reaction, my friends, is the reason that we can breathe. Brief interjection. Next time someone says that they don't like it when there are chemicals in their food, please remind them that all life is made of chemicals, and would they please stop pretending that the word chemical is somehow a synonym for carcinogen? Because, I mean, think about how chlorophyll feels when you say that. It spends all of its time and energy creating the air we breathe, and we're like, ew, chemicals are so gross. Now remember, all energized electrons from PS2 have been picked up by electron carriers and now are being transported to the second protein complex, the cytochrome complex. This little guy does two things. One, it serves as an intermediary between PS2 and PS1, and two, it uses a little bit of that energy from the electron to pump another proton into the thylakoid. So the thylakoid's starting to fill up with protons. We've created some by splitting water, and we moved one in using the cytochrome complex, but why are we doing this? Well, basically what we're doing is charging the thylakoid like a battery. By pumping the thylakoid full of protons, we're creating a concentration gradient. The protons then naturally want to get the heck away from each other, and so they push their way through an enzyme straddling the thylakoid membrane called ATP synthase. And that enzyme uses that energy to pack an inorganic phosphate onto ADP, making ATP the big daddy of cellular energy. All of this moving along the electron transport chain requires energy, and as you might expect, electrons are entering lower and lower energy states as we move along. This makes sense when you think about it. It's been a long while since those photons zapped us. We've been pumping hydrogen ions to create ATP, it's splitting water and jumping onto different molecules. I'm tired just talking about it. Luckily, as 450 million years of evolution would have it, our electron is now about to get re-energized upon delivery to photosystem 1. So PS1 is a similar mix of proteins and chlorophyll molecules that we saw in PS2, but with some different products. After a couple of photons re-excite a couple of electrons, the electrons pop off and hitch a ride onto another electron carrier. This time, all of that energy will be used to help make NADPH, which, like ATP, exists solely to carry energy around. Here, yet another enzyme helps combine 2 electrons and 1 hydrogen ion with a little something called NADP+. As you may recall from our recent talk about respiration, there are these sort of distant cousins of B vitamins that are crucial to energy conversion. In photosynthesis, it's NADP+, and when it takes on those 2 electrons and 1 hydrogen ion, it becomes NADPH. So what we're left with now, after the light-dependent reactions, is chemical energy in the form of ATPs and NADPHs. And also, of course, we should not forget the most useful, useless byproduct in the history of useless byproducts, oxygen. If anybody needs a potty break, now would be a good time, or if you want to go rewatch that rather long and complicated bit about light-dependent reactions, go ahead and do that. It's not simple, and it's not going to get any simpler from here. Because now we are moving along to the Calvin cycle. The Calvin cycle is sometimes called the dark reactions, which is kind of a misnomer because they generally don't occur in the dark, they occur in the day along with the rest of the reactions. But they don't require energy from photons, so it's more proper to say light-independent reactions. Or if you're feeling non-descriptive, just say stage 2. Stage 2 is all about using the energy from those ATPs and NADPHs that we created in stage 1 to produce something that's actually useful for the plant. The Calvin cycle begins in the stroma, or the empty space inside of the chloroplast, if you remember correctly, and this phase is called carbon fixation because, yeah, yeah, we're about to fix a CO2 molecule onto our starting point, ribulose bisphosphate, or RUBP, which is always around in the chloroplast because not only is it the starting point of the Calvin cycle, it's also the end point, which is why it's a cycle. CO2 is fixed to RUBP with the help of an enzyme called ribulose 1,5-bisphosphate carboxylase oxidase, which we generally shorten to Ribisco. I'm in the chair again. Excellent. This time for a biography of Ribisco. Once upon a time, a one-celled organism was like, man, I need more carbon so I can make more little me's so I can take over the whole world. Luckily for that little organism, there was a lot of CO2 in the atmosphere, and so it evolved an enzyme that could suck up that CO2 and convert inorganic carbon into organic carbon. This enzyme was called Ribisco, and it wasn't particularly good at its job, but it was a heck of a lot better than just hoping to run into some chemically formed organic carbon. So the organism just made a ton of it to make up for how bad it was. Not only did the little plant stick with it, it took over the entire planet, rapidly becoming the dominant form of life. Slowly, through other reactions known as the light-dependent reactions, plants increased the amount of oxygen in the atmosphere. Ribisco, having been designed in a world with tiny amounts of oxygen in the atmosphere, started getting confused. As often as half of the time, Ribisco started slicing ribulose bisphosphate with oxygen instead of CO2, creating a toxic byproduct that plants had to deal with in creative and specialized ways. This byproduct, called phosphoglycolate, is believed to tinker with some enzyme functions, including some involved in the Calvin cycle. So plants have to make other enzymes that break it down into an amino acid, glycine, and some compounds that are actually useful to the Calvin cycle. But plants had already sort of gone all in on the Ribisco strategy, and to this day, they have to produce huge amounts of it. Scientists estimate that at any given time there are about 40 billion tons of Ribisco on the planet. And plants just deal with that toxic byproduct. Another example, my friends, of unintelligent design. Back to the cycle. So ribulose bisphosphate gets a CO2 slammed onto it, and then immediately the whole thing gets crazy unstable. The only way to regain stability is for this new 6-carbon chain to break apart, creating 2 molecules of 3-phosphoglycerate. And these are the first stable products of the Calvin cycle. For reasons that will become clear in a moment, we're actually going to do this to 3 molecules of RUBP. Now we enter the second phase. Reduction. Here, we need some energy, so some ATP slams a phosphate group onto the 3-phosphoglycerate, and then NADPH pops some electrons on, and voila! We have 2 molecules of glyceraldehyde 3-phosphate, or G3P. This is a high-energy, 3-carbon compound that plants can convert into pretty much any carbohydrate, like glucose for short-term energy storage, cellulose for structure, starch for long-term storage. And because of this, G3P is considered the ultimate product of photosynthesis. However, unfortunately, this is not the end. We need 5 G3Ps to regenerate the 3 RUBPs that we started with. We also need 9 molecules of ATP and 6 molecules of an ADPH. So with all of these chemical reactions, all of this chemical energy, we can convert 3 RUBPs into 6 G3Ps, but only one of those G3Ps gets to leave the cycle. The other G3Ps, of course, being needed to regenerate the original 3 ribulose bisphosphates. That regeneration is the last phase of the Calvin cycle. And that is how plants turn sunlight, water, and carbon dioxide into every living thing you've ever talked to, played with, climbed on, loved, hated, or eaten. Not bad, plants. And I hope you understand if you don't. Not only do we have some selected references below that you can check out, but of course, you can go rewatch anything that you didn't get, and hopefully, upon review, will make a little bit more sense. Thank you for watching. If you have questions, please leave them down in the comments below.